Hello guys, in today's lecture we are going to talk about ETC cycle, or it's also called oxidative phosphorylation. It is the last step of cellular respiration. So let's rewind the previous steps or first steps before these steps. So first step is glycolysis. By glycolysis, glucose is converted into 2-pyruvic acid. This is the first step of cellular respiration. The second step, the pyruvate has a many fates, but if it goes into the aerobic organism, then it convert into acetyl CoA by PDH complex. This is the second step to formation of acetyl CoA. Now this acetyl CoA is undergoes the TCA cycle. Via these three steps, the reducing power called NADH and FADH2 is produced, which is not an actual currency of energy, but it is the form of conserved energy which can oxidize and form energy in ETC cycle, electron transport chain cycle. So let's start. So what happened? It is a series of electron carrier and complexes. Four complexes there. First of all, I just forget to told you that ETC cycle is run in mitochondria, especially in mitochondrial inner membrane, because the complexes are present which is embedded into mitochondrial intermembrane. So the series of electron carrier and complexes, the hydrogen ion, the H plus proton meets with the final terminal electron acceptor called O2 and form H2O with the proton gradient. By this gradient, ATP synthesis form ATP. This is the overall of ETC cycles. Now let's go in detail. Electrons are funneled into the universal electron acceptors like NAD, H and FADH. These two are the universal electron acceptor by their dehydrogenases enzyme. Dehydrogenases. Now the NAD link and FAD link. So first talk about NAD link. NAD link dehydrogenase remove 2 electron from the substrate. So it removes 2 electron or 2 electron, 2 H plus. 1 electron or 1 H plus is done by the NADH. So NAD form into NADH by getting 1 hydrogen. But 1 hydrogen or 1 electron is donate into the or release into the medium. Now electron is transferred by 3 ways: direct transfer into the electron carriers like Fe plus 2 to Fe plus 3. Second one as a H plus atom, and third one is as a hydride ion. Like this, you have 2 electron. this t-type electron transfer into the ETC cycles. Now there are rest of this other electron carriers are also present, which carries electron, not conserve electron like this. Electron carriers, mainly 3 electron carriers are present in the ETC cycle. 3 electron carriers are present into ETC cycle. It is a hydrophobic quinone called ubiquinone. The second one is cytochrome, which are for the 3 types A, B and C. The third one is a iron sulfur protein or carrier. Now these two carriers, cytochrome and iron sulfur protein, contain iron containing carriers. And this is not iron containing, it is a hydrophobic carrier. So let's about ubiquinone. Ubiquinone, it is a one type of benzoquinone which have a isoprenyl side chain in its structure. Now it is also known as a coenzyme Q, and also simply by Q or by quinone. Now ubiquinone, by getting one electron, this is the ubiquinone, by accepting one electron, it converts into semi-quinol radical. Sorry, semi-quinone radical called QH star. And by getting 2 electrons, it completely reduces into ubiquinol. Which is highly stable, and it is not a stable, it is a dangerous. Now the second one is a cytochrome. This is the one of the last electron carrier. Through this, the electron, one electron is transferred to the complex 4 and then to the oxygen. So cytochrome has a high UV visible rays absorption spectrum. So by this spectrum, we distinguish the 3 types of cytochromes, which is the A, B and C. Now A is a spectrum band nearer 600 nanometers. Now B has a 560 nanometers and C is a 550 nanometer nearer band when it is exposed to the UV visible absorption light. So these are the iron containing, especially heme. So heme group of A and B, it is not a directly covalently connected with their associated protein, but cytochrome C is a covalently attached to his nearer or connected protein. This is the main difference between ABC. Now the last and final is the iron sulfur protein. The iron is present or connected with the either inorganic sulfur or with the proteinic sulfur with the cysteine amino acid, because we all know cysteine amino acid and methionine amino acid containing sulfur. So by directly cysteine or by the inorganic solution or both. It can be both. So the structure, if we see the structure, then the iron Fe is be found in the center and surround with the 4 sulfur acid sulfur, and here the cysteine connected. This is a single, and if we draw about the two different or two connected Fe, then it looks like sulfur, with the cysteine residue. Now this is the common alpha sulfur protein, but if we talk about the risque iron sulfur protein, then this is the main difference between the simple one and risque. It is given a name by scientist which found John Risque. To given in the honor of the scientist to give a name risque iron sulfur protein. He bound with the two histine residues rather than cysteine residues. Now let's go main part of BTC cycle. The electron is passed through the series of 4 different electron containing complexes. The complexes now start let's talk about the first complex. It is a NADH dehydrogenase. This complex also known as. So in this complex it looks like this is the inner membrane. Inside this is positive side. So this is the first complex called NADH dehydrogenase. Here it is look like a inverted L or like a 7. So it is containing two arms, membrane bound arm, and the extragonadical matrix because it's present in the matrix, because this is the inner membrane of the mitochondria. So into the extended arm the FMN is present. FMN is taken electron from the NADH to reduce oxidize NADH into NAD. Now FMN through series of 6 FES domains, the electron is finally meet with the quinone, which is present into last. Now the quinone is reduced into QH2. This happen in NADH dehydrogenase complex. This is the first complex. It contains 42 polypeptides. The second complex is known as succinate dehydrogenase. If we draw the complex, it must look like this is the inner membrane. Divided into 4 regions called A, B, C and D. So this is the complex 2, succinate dehydrogenase. Here it divided into 4 different regions A, B, C, D. The C and D present in the intermembrane. or the membrane bound, and the B and A, it is extended like the complex 1 extended arm into the matrix. Now succinate is converted into fumarate by giving to electron, because in second complex the electron donor is a succinate. In first was the NADH into the FAD. In previous FMN was present, and here FAD is present. To FAD, first it goes to the succinate binding site, you bind and give electron to FAD, and through the 3 FES complex, it's give electron to the Q to form a QH2. Here the different in D portion, the heme is present. Heme is not directly connected into the electron transfer, but it is the reduce the frequency of leak electron. Now what is leak electron? So during this reaction, if any electron from succinate is leaked and connected with the oxygen, and oxygen is converted into ROS, reactive oxygen species, like nascent oxygen, and this is the very harmful effect containing. This is done very harmful. So if this form, then tissue is damaged. So in point mutation containing humans, disease cause paraganglioma. Paraganglioma. We have a mutation into the second complex, especially into the D region heme. So that's why they have a large proportional concentration of ROS in their body. That's why their tissue is very much damaged. This is the succinate dehydrated, the second complex. Now the third complex, it is a ubiquinone cytochrome oxido reductase. It is a complex. I mean, it is very complex to study. So let me draw first. So this is the multi-complex complex. So here into the left side, and this is the right side. So first quinone is enter into the cytochrome B. This is cytochrome C. This is cytochrome C1. This is FEX complex. So into the cytochrome B, and by getting one electron, it convert into semi-quinone radical. Simultaneously, in the second complex right side, the semi-radical quinone is converted into QH2 and split outside the complex by getting a 2 proton from negative side, inner side. So between this, here the in cytochrome B, the QH2 is also entered, and QH2 is dissociated into 2 different electrons. Now the cytochrome C contain only one or transfer only one electron, so it will take one electron from QH2, but the other electron is taken by Q to form a back, or to form a QH2, or form a direct Q2 semi-quinone. So through series of 2 carriers or 2 different regions, heme BL and heme BH. So this QH2 is dissociated into 2 different. So let me draw here QH2 is dissociated into two electrons. Here the first electrons is driven into the near side of the Q. Nearer Q will take the electron first. Now second electron is through FES goes into cytochrome C1 and then cytochrome C. During this, the two proton splits outside the complex into intermembrane space. I forget to tell you, in first complex 4 proton also goes or pump outside. So that's why this outer side or space side become positive and thus matrix side become negative. Simultaneously here the QS2 also enter and same thing is happen, because at a time cytochrome C can transfer only one electron, so two different complexes there to transfer the two different electrons or two electrons simultaneously. This is also known as the Q cycle, because here ubiquinon to cytochrome electron is transferred. This is the complex 3, ubiquinon cytochrome oxidoreductase. Now the final complex 4. Here the electron is flowed to cytochrome 2, the final electron acceptor O2. So it's look like it divided into 3 different regions: subunit 2, subunit 1 and subunit 3. Here the 4 cytochrome C electron in the first CuA portion of the complex or 4 into the subunit 2. Now through series of reaction into complex 1, the 2 heme A3 and heme A is present. So through this it funnels into CuB, and through CuB it's transferred into the O2 to H2O, and during this 4 protons also funnel outside, and the O2 is get finally converted into H2O. This is the last step of the ETC cycle and the cellular respiration. So during these 4 complexes, the complex 1, complex 3 and complex 4 are pumped 4 hydrogen outside. So that's why this inner matrix side is into the negative and the intermembrane space is become positive. So gradient is generated. Through gradient the enzyme called ATP synthase, which is located near the complex 4 like this. Moves certain degree and form a ADP to ATP, and also pumps hydrogen into the matrix to balance the proton gradient. So this is all about ETC cycle. I hope you enjoyed and learned the video. Stay connected with Ravi's Microbiology. Thank you.